I can't see hands raised. I can't see the chat. So if there are questions, either Tochukwu helps to read them or you ask.
Okay, so we have none for now in the chat.
Okay.
But if you have any question, just unmute and ask now, or you drop this box.
Okay. So let's start with the genomic, with the genetic code. So when Watson and Crick determined the structure of DNA, one of the things they were not sure about, or in fact they had no idea, is how the information in DNA encodes the ability of a cell to build itself and perform its functions. So how is it that the DNA is red? You remember that at the end of their paper, they did say that, look, the mere structure of the DNA gives us a hint as to how it's copied, but not how it's read. How do we make sense of it? Now, but they were at least prescient enough to know that there was some sort of code, okay? And in a code, essentially, one thing means another thing. So what we have up here is not the genetic code. This is the Morse code. Okay, some of us in school, we learned to either whistle or flash SOS on the Morse code. Every letter has either a code of long or short symbols. So you can say S by doing 3 short flashes, and then you have S. And anybody else who knows the Morse code, when they see 3 short flashes will know, oh, somebody's trying to say S. So there was clearly a code with rules, but with the genetic code, nobody knew what those rules were. And Crick had actually some interesting ideas about what the code would look like, which largely turned out to be true. So he published a paper just a couple of years after the structure of DNA paper that actually outlined what he thought the code would look like, even though he didn't know the code. So the first thing that was in that paper was that he thought that so first of all, remember, there are 4 bases in DNA, and there are 20 amino acids that specify a protein in cells. And essentially what Crick was outlining is how that DNA could specify information in proteins that function. So he said, first of all, he thought that 3 bases would code for an amino acid. One of the reasons why he thought that is that in the experiment that was done by, let me get it from last time. There we go, Janowski's experiment. When mutations were made, if insertions and deletions were made, they changed the entire amino acid sequence. If you made 1 insertion or 1 deletion, if you just changed if you just changed the base, you changed an amino acid. But if you inserted a base or you removed a base, you changed the complete downstream sequence. If you did that with 2 bases, you added in 2 bases or you took away 2 bases, the same thing happened. You changed the entire downstream sequence. But when you put in 3 bases, the sequence remained the same. So Crick thought that multiples of 3 might specify an amino acid. And even before Crick said this, most people were convinced that the code could not be based on 2 nucleotides. Why is that? Why is it that it could not be multiples of 2? Other than the fact that if you insert 2 or you remove 2, the entire sequence changes. Some of you have done this in class. Why is it that the code, the genetic code cannot be 2 bases specifies an amino acid? So, Trick, any hands up, please?
Okay, none for now.
Okay, let's wait.
Yeah, Victor, you can go ahead.
All right. So I think if there are two, we would have just 16 amino acids being coded for, which is not up to 20.
Absolutely, that's correct. How did you get 16?
4 to the power of 2.
Okay, fantastic. So if the code was in twos, we would not have enough information to code for 20 amino acids. So that's why he thought it would be threes. Theoretically, it could be fours. If it were fours, you would have enough to code for more than the existing amino acids. But generally in biology, we like to start with a hypothesis that suggests that the simplest solution is the solution. And then we will disprove that before going on to anything more complex. Okay? So he was thinking, well, maybe amino acids in threes bases in threes specify amino acids. You can imagine that in a code, it's possible that one group of 3 next to another group of 3 specifies one amino acid next to another group of amino acids, or that the code could be overlapping, as in 1, 2, 3 could specify one amino acid, 2, 3, 4 would specify another, 3, 4, 5, and so on. Okay, but Crick had read very carefully the existing literature, and he realized that people who had done research with tobacco mosaic virus found that if you change one base, you either change no amino acids or you change one amino acid. You never change more than one. So if it were overlapping, if we changed base 2, then we should change the amino acid for 1, 2, 3, and 2, 3, 4. Okay? So he felt it was non-overlapping. The other thing that Crick thought was that the code would would be read from a fixed starting point. Okay? Because if there was no fixed starting point, then there must be punctuation. Otherwise, the cell would not know how to read the information. Okay, if we write a sentence with no spaces, no punctuation, if we have a starting point, we might be able to figure out what that sentence means. But if we're going to start at a random point, it'll be a lot harder. Okay. And then he thought that the code was probably degenerate. And that is that one amino acid, there would be multiple groups of 3 that could code for it. And the reason goes back to Emeka's comment about twos specifying only 16 amino acids, but it turns out that threes would specify 64 using the same formula that he used. And we only have 20. So that means some specify more than one. Okay. Any questions so far? Okay, so let's go into how experimentally Crick's hypothesis were shown to be true. So Crick is a theoretical biologist. Okay, before this, the experiment he's done that he's famous for is building DNA from models. And now he's just laying out ideas about what he thinks the genetic code should look like. But he actually is not an experimentalist. Okay. So, I have my slides in the wrong order. So let me find it. Here we are. So the specific language in the code was cracked by researchers at the US National Institute of Health, led by Nuremberg. Okay. And what he did was quite clever. Investigators before him, including the likes of Janowski, had figured out how to get DNA transcribed in vitro in a test tube. Sorry, not DNA, RNA. Sorry, I missed that again. Had figured out how to get RNA translated in a test tube. So if you put RNA in a test tube with all the right buffers and so on, and you put ribosomes that you have purified from an E. coli cell, you can actually get the ribosome to translate the RNA. You would need to provide in the medium all the 20 amino acids. So Nuremberg thought, well, why don't I just provide an RNA that contains only one base? And so the base he picked was U. You know, in DNA, we don't have uracil, we have T instead. Okay, but in RNA, we have uracil. So he made a strand of RNA that contained only U. And then he put this in a test tube with ribosomes and amino acids and so on, and then harvested the polypeptide that was made and analyzed the sequence of that polypeptide. And the polypeptide was entirely polyphenylalanine. So that meant that every single base in that polypeptide was phenylalanine. And then he had then figured out that a triplet sequence of UUU encoded for phenylalanine. And so they did the same thing for AAA, CCC, GGG. And then they had 4 letters of the code. The next thing they did was quite clever. They started mixing different amino acids, but doing it in a systematic way. First of all, starting with 2s. If you make a strand of RNA that alternates C's and A's, then you end up with alternate codons of CAC and ACA. And when they did this, they got a peptide chain that was threonine and histidine alternating. And so they did this again for all the combinations of the bases. And by then they were halfway through the genetic code. And then they made more complicated mixtures. And in the end, they were able to figure out what every amino acid was coded by. So for example, UUU codes phenylalanine. We had seen that before. It turns out that phenylalanine is one of the many examples where there's degeneration, that where the code is degenerate. So UUU encodes phenylalanine. So does UUC. Okay. And you'll see that there are some amino acids that almost 6 codons can code them. Leucine is a popular one. Arginine is a popular one. And there are other amino acids that there are only 2. And there are 2 amino acids, methionine and tryptophan, that are only encoded by 1 codon. They were also able to find that there are 3 codons that do not encode any amino acid. These are referred to as nonsense codons because what they encode is a full stop, the end of a protein. So they're often referred to as stop codons. So in a gene, we typically have start codons, a start codon, which is almost always, not always, but almost always methionine, and one of three stop codons at the other end, and then in between, whatever codons we have will specify what the protein looks like. This is a slide I had before. I'll just take you back there. So a gene looks like this. This would be a strand of DNA that would be transcribed to RNA. Ahead of the start of the gene, there would be a regulatory region. The regulatory region contains information that allows the gene to be turned on. That is for transcription, for permission to transcribe to be given. Usually what is here are sequences that allow RNA polymerase, the enzyme that transcribes DNA into RNA, to bind. Okay, so RNA polymerase, one of the subunits known as sigma, its job is to bind upstream of a gene so that RNA polymerase can then read that gene. Then there's a transcribe region, and at the beginning or very close to the beginning of the transcribe region, there'll be a start codon, usually methionine, ATG, and then at the end, there will be a stop codon and a transcriptional terminator. So this is a kind of simplified structure of what a gene looks like. So genes are they're encoded on the DNA. RNA polymerase reads the DNA and makes, through transcription, an RNA copy of one of the strands of the DNA. We call this transcription because transcribing is copying. And the language of DNA and the language of RNA are very similar. The only difference is that everywhere there's a T in the DNA, there'll be a U in the RNA. And then where there is deoxyribose in the DNA, there'll be ribose in the RNA. But by and large, the language is the same. When we're going from RNA to protein, the language changes. The nucleotide language that you have in DNA and RNA is different from the amino acid language that you have in the protein. So that means that the RNA strand will have to be translated into protein. Sometimes this scheme I'm showing you on the slide is referred to as the central dogma. DNA goes to RNA, RNA goes to protein. And we're also told that you cannot go backwards. You cannot make RNA from protein, and normally you cannot make DNA from RNA. This is true in most cells. The exception is that some viruses have what is known as reverse transcriptase. Retroviruses are your example, and they can actually transcribe RNA into DNA. But by and large, most cells, including yours and mine, can only go in one direction. In a eukaryotic cell, the DNA is in a nucleus. So the nucleus here is on the top left hand of the screen, and the nuclear membrane is here. And the DNA stays in the nucleus. It only comes out of the nucleus when the nucleus is dissolved for the cell to reproduce. And then when the new daughter cells are born, a new nuclear membrane is built. Okay? So in the nucleus, that is where RNA polymerase transcribes the DNA into RNA. And then the RNA is processed in eukaryotes. This processing involves removal of sequences that are not going to be translated. This is referred to as splicing, and putting a cap and a tail on the RNA. Once that is done, then the RNA is exported from the nucleus through a nuclear pore. And then that RNA is then in the cytoplasm loaded onto a ribosome, which can then translate it. The ribosome will use tRNAs to read the codons in threes and bring in the corresponding amino acid. And then the amino acids will be joined together by forming peptide bonds in the ribosome. That's how the central dogma operates in real life. Any questions? So this is the genetic code in a manner that a beginner might want to read it. Most of us who are biologists do not read the genetic code this way. Bear in mind that each amino acid has a single letter code and 3-letter code. After you've worked with the amino acid sequences for a while, you actually get to know what these are. Okay? I don't usually advocate people memorizing them. Eventually, you'll learn them. It's like learning a language. Okay? I never advocate anybody memorizing the genetic code because you can get it. It's online, it's on my slides. If you go to the Android store or the other one for iPhones, you can download yourself a free genetic code app. When you do, you'll probably be looking at the genetic code this way. Same information on the other slide, but organized differently. I'd like to take us through that, but I think there might be a question because I heard a noise. So is there something in the chat?
Yes, there is a question. So you can unmute yourself and go ahead, Kehinde.
Okay, thank you, Ma. So I want to ask that. Hello, can you hear me, Ma?
Yes.
Okay, Ma. Thank you, Ma. I want to ask that you were saying that the DNA sequence that we have the coding and nonsense coding, which is coding for the stop codon and the start codon, and you also mentioned that there are some sequence base of sequences that are before the start codon and after the stop codon. So these sequences, are they not also in the order in the are they not also bases such that they are also represented by all these UC, GC that they will be coding for another kind of protein or something? I don't get that. You said the those sequences before the start codon is performing a kind of regulatory activities whereby the polymerase will be binding to it. So my question is that does it not contain sequences, or why is it coding for any protein, or maybe you can just shed more light on that. Thank you, ma'am.
Okay, thanks for the question. Not all nucleotide sequence codes. In humans, I forget the proportion, but a very small proportion of the DNA in humans is coding. Most of the DNA in humans is never translated into protein. When this was first discovered, the DNA that was never translated to protein was referred to as junk DNA. That's a bad thing to call it, because if I delete all your junk human DNA, you are no longer human. So the junk DNA has a function. The only thing is that its function is not coding for protein. Some of it is doing what I described. It's allowing regulation of things that are coding. Some of it has a whole range of other functions, some of which are known, some of which are not known. Okay? In a few minutes, we'll actually look at some DNA, and then you get a better sense of what that means. But just bear in mind that we're very excited and interested in the DNA that codes because the function of that DNA is very obvious to us. But that doesn't mean that DNA that doesn't code is useless. Okay, even stop codons. There's the old language that we used to call them nonsense codons. Now we know stopping is very, very important. So stop codons are critical. even though originally they were seen as nonsense because they were terminating the translation of a protein. Okay, it's a good question. Any other questions?
None for now.
Okay, thank
Okay,
you.
I want to. Sorry, can I ask a question? Just this last question.
Yeah.
Okay, I want to ask. You mentioned that most of the uh, sequences in a, let's say, in a human genome, most of only few of them is actually coding for a protein. So I want to ask that, and you said that if there is only a kind of insertion of one or two base sequence, that is when there can be a kind of a genetic change or defect. So now, if there is
Okay, I don't know whether he's gone or he's muted, but I think I get the question. I was saying that if you insert one nucleotide into coding sequence, you change the code. This is true, but that is about coding sequence. The way you break other kinds of sequence is different. Okay, so adding a nucleotide or removing a nucleotide from non-coding sequence will have a different consequence than coding sequence. So right now we're pretending we're in the 1960s and we're talking about coding sequence. But as you get more adept, you too will start to take more interest in non-coding sequence. I work with E. coli in bacteria. Not all sequence is coding, but a lot more is coding. In bacteria, I think roughly 70 to 80% of the sequence is coding, but there's still non-coding sequence. So right now because we're looking at the genetic code, we're looking at coding sequence. But as I mentioned, in a minute, we will go, not in a minute, but in a few minutes, we will go and look at real sequence. And then we can hopefully explain or give you a visual view of how some parts are coding, some parts are not coding, and so on. Okay?
oh, I get it now. Thank you very much, man.
Okay, so this is the genetic code as we often write it. The first letter we list here. The second letter, so here being on a kind of y-axis, okay? And then the third letter the second letter along the top, and the third letter at the far right. So for example, if I wanted to know what does UUU encode, I would say first position U, second position U, third position U. The amino acid encoded by UUU is phenylalanine. 3-letter code for phenylalanine is PHE. 1-letter code is F. If you see how I read it, you'll also see that UUC also encodes phenylalanine. Whereas UUA encodes leucine, and UUG also encodes leucine. Is there anybody not clear about how to read this table? Okay, I didn't hear anything, so I'm assuming it's straightforward. Okay, if I want to know what AUG encodes, I will say first position A, so I'm in this general area. Second position U, third position G, and I can see it encodes methionine. As I mentioned earlier, methionine is only encoded by one codon, and that codon is the most common start codon. So most peptides made in a cell begin with methionine. Now, if you look at this code, one of the things you will notice is that where you have some degeneracy, that is where you have multiple codons encoding the same amino acid. Very often, not all the time, but very often, that amino acid is in the same section. So for example, if the first position is C and the second position is U, whatever the third position is, you're going to get a leucine. If the first position is C, second position is C, whatever the third position is, you're getting a proline. If the first position is G, second position is C, whatever the third position is, you're getting an alanine. Now, interestingly, when mutations happen, the likelihood that that mutation will have a consequence depends on where that mutation takes place. If you have a mutation in the third letter of a codon, you're less likely to see a consequence than if you have a mutation in the first letter. If you look at the genetic code, it immediately becomes clear why that is the case. If I change the third letter, sometimes I change the amino acid. For example, if I changed AAU to AAA, I have changed an N to a K. But most of the time, if I change the first amino acid, I change the letter. Sometimes if I change the third, I change the letter, but most of the time, I do not. Okay? So if I change AAU to AAC, I still have asparagine. I didn't change it. It's only if I go to AAA or AAG, then I end up with lysine. And interestingly, what's even more interesting than what I just said is that both asparagine and lysine are basic. They're basic amino acids. So that means that even if I change that third nucleotide in the codon, and I do get a change, I will change one basic amino acid to another. If I change GAU to GAC, there's no change. But if I change it to GAA, I change an aspartic acid to a glutamic acid. And essentially, I've changed an acidic residue to another acidic residue. So the genetic code, it was not compiled randomly. You can see that related things are in related sections. See where all the stop codons are? There are only 2 tyrosine codons, and they are together. So a lot of people actually just look at the code and study it and understand more about biology just by looking at it. I'm not going to ask you to do that. I mean, why reinvent the wheel? If somebody else has done that, you may not have to spend 3 weeks staring at the genetic code. Instead, I'll send you an article that allows you to see the beauty of the genetic code and to see some of these things that I've mentioned. Now, one of the things I want to point out is that when we're looking at sequence, it's important to understand which sequence we're looking at and to orient ourselves accordingly. Just a minute. The homework picture. Black and white. Thank you. Okay, so it's important to think about which molecule we're looking at and what orientation it is. The DNA strands, if you remember when we looked at Watson and Crick's model, they are anti-parallel. So if the top strand is running 5' to 3' in this direction, the bottom strand will be running the other direction. and the two strands will be complementary in that every T in that strand will be paired to an A in the other strand. Every C in that strand will be paired to a G in the other strand. Okay? So that means that if I'm looking at the top strand, often referred to as the coding strand, my orientation will be going from left to right, as if I'm reading English, and my DNA will be labeled 5' to 3' on the top strand. We call that the coding strand, and the messenger RNA that is obtained from that DNA will essentially be the same as the coding strand, except that the T's will be used. That means that when I look at the genetic code, the mRNA will be UUU for phenylalanine, and the coding strand for the DNA will be TTT. Okay? There is the complementary strand to the coding strand is the template that is used to transcribe the DNA into the RNA. RNA polymerase, the strands separate when transcription is taking place. RNA polymerase will sit on the template strand and copy it and then make something that is complementary to the coding strand. The tRNAs, if and when you do biochemistry, you learn how they function with the ribosome. But they bring in what is referred to as the anticodon into the ribosome that binds to the codon on the mRNA to bring in the amino acid. Okay, so the amino acid encoded by TTC or UUC will, its corresponding tRNA, the anticodon it will have will be AAG. And it will bring in this amino acid in this orientation. The DNA is going 5' to 3'. Janowski found that the protein will be going amino N terminal end, that's the amino end, to C terminal end, the carboxy end. So let's illustrate this with a cartoon. We have double-stranded DNA here. The top strand is the coding strand. Some people call it the sense strand. The bottom strand is the template strand or the non-coding strand. So when the strand separates, RNA will be made that is complementary to the template strand. Okay? And then that will be the mRNA that is then taken to a ribosome to be translated. Any questions?
Okay, so Abib, you can go ahead.
Okay, so my question is, so what determines which strand to use as the template?
The promoter sequence.
Okay.
The sequence ahead that will determine where RNA polymerase binds is what determines which strand will be used as a template. Where is Google Meet? Other questions? I'm
Sorry,
still
Matthew.
sharing.
Can you come again, ma? You said something I hear you very well.
Abib asked, what determines which strand will serve as a template? And I said it is the sequence ahead of the coding information that determines where RNA polymerase will bind. Okay, I guess I'm not sharing because I can see hands now. Omoto Shaw, kindly, you have a question?
Yes, yes, ma, I do. I want to ask that does any error occurs during the translation period, since the genetic sequences are inside the nucleus, and that's where we mostly see errors. But now, when the translation occurs in the ribosome, does error usually occur there? So I just want to ask. Thank you.
Sometimes. So transcription is a place where a lot of error occurs because RNA polymerase has no proofreading activity. But sometimes a wrong anticodon wobbles on the ribosome and brings in a wrong amino acid. Yes, if it does, then you have a wrong protein. But remember that both transcription and translational errors have less consequence because you can always retranscribe and make a new RNA. You can always retranslate and make a new protein. But replication is when you copy it and take it to a new cell, and then you don't have time, you don't have the opportunity to redo it. Other questions?
Okay, thank you, ma'am.
Yes,
Okay.
ma'am. So my question is this: can both strands serve as templates, and can both strands have the sequence for transcription regulation?
The answer is yes. And we're going to see that when I open some sequence for us to look at. Okay.
Okay. Okay.
Sorry, I'm just trying to open the file. Okay, then let me try and share. Okay, so if you're not on a computer, this is going to be hard to see. Okay, so you're going to need to zoom in. Okay, but we're going to be looking at some sequence on part of the screen here. Okay, now where I'm putting my mouse and highlighting in yellow, that is DNA sequence. Okay, I'm reading AGAGATTACGT. Can we see that?
Yes, ma'am.
Okay, lovely.
Yes. Ma'am.
So I'm reading a strand 5' in the 5' to 3' direction. The numbers here are just telling me how many nucleotides at each position so that I can keep track of where I am. Okay. Now, what I read to you is the top strand. If you look carefully, the A here, the AGA here is paired with TCT on the bottom strand. Can we see that?
Yes, ma'am.
Yes, ma'am.
So essentially, these two strands are double-stranded DNA. They're actually from the chromosome of a Salmonella strain that I'm studying with our Part 5 students right now. Okay, so this is DNA. One strand 5' to 3' going this way, and the other strand 5' to 3' the other way. Okay, and you can see the two strands are complementary. Okay. Now, if I go to the top strand and highlight the first three bases, what you can hopefully see is that the top line in the 3 lines above the DNA strand and R highlighted. Okay, so I highlighted AGA and I saw an R. If I look at my genetic code, I can see that A, G, A is arginine or R. Hopefully now you can see why I said get yourself a genetic code, okay? Have one handy. I have my screen in front of me and I'm looking at sequence. So I'm looking at the genetic code on my phone to be able to tell you that AGA is arginine. I never memorized that AGA was arginine, nor do I ever plan to do so. Okay. All right. Now, next to AGA, the very next codon is GAT. And you can see GAT is D. We're still on the top strand. And then after that, I have TAC, and that is Y. So essentially, if I start translating the top strand from the very beginning of this file, I'm getting a translation that is up here. If instead of starting at position 1, I start at position 2, that is, I ignore this first A, and the first codon I read is GAG. GAG translates to an E, and that's what I have up here. After that, now after GAG on positions 2, 3, 4, my next codons will be 5, 6, 7, ATT, isoleucine, ACG, threonine. Notice that when I started on the second nucleotide, I began to translate amino acids that are written on the second line above the strand. Okay? Now, if I start on the third nucleotide, which is A, and my first codon is AGA, that is arginine, TTA, leucine, CGT, arginine, and so on. I'm now on the third row of amino acids. So I have a question for you. If I start the translation on nucleotide 4, which of the lines of amino acids am I going to be reading? Is there anybody that can answer that question? Does the question make sense?
The first line, ma.
Okay, who is
The
answering?
the first reading frame.
Grace.
Grace, okay. Are you in part 5?
No, ma.
Okay. I was going to say I didn't want anybody in part 5 answering the question because we did this a couple of weeks ago. Okay. So, yes, Grace, if I start on 4, I start reading on the first line. This is true. Okay. So here's 4. And I'm reading everything. If I start 4, this is the first codon. If I go to the next codon, I'm reading everything except the first amino acid. Okay. So essentially, this strand has got 3 possible reading frames. This is the first one, this is the second one, this is the third one. And which one I'm reading depends on which base I'm starting on. If I start on the first or the fourth or the 7th, I'll be reading the top. If I start on the second or the 5th, I'll be reading the second one. And if I start on the 3rd or on the 6th, I'll be reading the 3rd one and so on. Okay? Now, on your screen, you can see we have almost 200 nucleotides there. If you look at the numbering, it's a chromosome of Salmonella. It's a lot bigger than that. It's just that the screen can only fit in so many things. I can actually scroll down the sequence and keep moving. And you can see the numbers going up and going up and going up and going up and going up. It's a very large file, okay? To the other end of the sequence. And I got to the end here. And what you can see here is that I have something like 4.8 million nucleotides in that chromosome. Okay? sorry, base pairs, because one strand is 4.8 and the other strand is 4.8. Okay. Now, why I brought you here is I would like to read the bottom strand. And to read the bottom strand, I have to move in the opposite direction as if I'm reading Arabic. Because DNA is always read in the 5' to 3' direction. And if the top strand is in that direction, the other strand will be anti-parallel. So I start from the bottom. My first codon is now TAT if I'm reading in the 5' to 3' direction. TAT translates to a tyrosine, which is Y. My next codon is also TAT, which also translates to a tyrosine. And my next codon is TTT, which you remember as UUU, phenylalanine. Okay? Notice that when I started at the end, I'm reading the very bottom strand below the sequence. Before we were reading the top strand and we were reading frames above the sequence. For the bottom strand, when I started with the last nucleotide, I started at the bottom. If I start at the second to the last nucleotide, my first codon then will be ATT isoleucine, followed by ATT isoleucine, followed by TTA leucine. Okay? And if I start on the third codon, then my first codon is TTA leucine, TTT phenylalanine, and TAA, which gives me a symbol. What's the symbol? What's the symbol? What's,
Ash.
yeah, what's the hash indicating?
A
The stop
subcodon.
codon.
Stop codon. So the letters you have here are the single acid amino acid code, and the 3 stop codons are indicated by symbols, hashtag, plus, and I think asterisk. Okay? So hopefully you can see that in one double-stranded molecule of DNA, there are 6 possible reading frames. 3 of them are on the top strand, going from left to right, as if I'm reading English, and 3 on the bottom strand, going right to left, as if I'm reading Arabic. So that for every double-stranded molecule of DNA, there are 6 possible reading frames. Now, this goes to the question that was asked. Where would the genes actually be? Where would the coding sequence be? It turns out that the coding sequence could be on either strand. It just depends on where the promoters are, where the stop codons are, and where the start codons are. Okay? So, this sequence that you've been looking at, what I'm going to do now is zoom out. Okay, you've been looking at letters. Okay, we've been looking at T's and A's and G's and C's, and we've also been looking at amino acid sequences, F and R and so on. Okay, now I'm going to zoom out so far that you'll not be able to see the letters. Okay, let's do it slowly so that you don't think I'm working magic or something. Okay, so I zoom out slowly. You can't see the letters, but you can see the numbering, and you can know that I'm fitting more and more sequence into my screen. It's just that you can't see the letters anymore. And then hopefully you can see, if you look above where we're looking, that we have something like that as well. So what you have above is the same as what you have below. It was just zoomed out more. So let me bring our letters back. Letters are back here. Above here, you can't see any letters. But let me take us all the way to the beginning again. Long journey, long journey, long journey. Long journey to the beginning. At the beginning, you can see that on the bottom, I have the first 20 letters, whereas on top, I have everything going from the beginning to 800 in the same space. So obviously, I can't see the individual letters. But I can see gray for where the DNA sequence is represented. And I can also see 3 rows above and 3 rows below. Okay? And there's some lines on these 3 rows. What these lines are all the stop codons. All the stop codons. So, well, there's no need for me to do that. I've told you what they are. They are all stop codons. So as you can see, in from the beginning to about 2000, maybe 700, there are lots of stop codons on all 3 reading frames of the bottom strand and on 2 reading frames of the top strand, but no stop codon on the top reading frame. So these 5 reading frames, starting from the bottom, 1, 2, 3, 4, 5, have been closed by stop codons. Whereas the top reading frame is open. There are no stop codons in this region. In other regions, there are stop codons. There are no stop codons in this region. So we refer to this as being an open reading frame. This reading frame is closed. This reading frame is closed. This reading frame is closed. This one is closed. This one is closed. But this top one is an open reading frame. It is open because it contains no stop codons. Any questions? Okay.
Okay, so someone has one.
Okay, what's the question?
Chidima, you can go ahead and unmute.
Yes, thank you very much. I wanted to ask. I understand that the open reading frames do not have any stop codons, so that's what the black lines help us signify. But is there anything that helps us signify start codons as well? Because I remember you said that there would be some codons that do not code for proteins, which are between start and stop codons.
Yeah, there's some sequence that does not code for proteins, exactly. So remember that most start codons are methionines, okay? It's possible to have methionines inside of the sequence of a protein, and then you'll also have them at the start, okay? So on this region that you're looking at now, we've marked the stop codons with lines. Every black line here is a stop codon. Right now, I'm just going to take the stop codons away. Okay, I'm going to unmark them. They're still there, but I'm just going to remove the ability to see them. Okay, I've removed them all. Okay, now I'm going to mark the start codons. Okay, those are the start codons. They are pink. Okay, and now I'm going to bring the stop codons back. As you can see, there are stop and start codons scattered everywhere. Notice that the open reading frame that we were studying, that had no stop codons within it, has a large number of methionines within it. There are methionines close to the beginning, there are methionines at the end, there are many in the middle. Okay? For something to be a gene, it must first of all be an open reading frame. Okay? That means there must be no stop codon within it. It must also have a start codon at the beginning. Start codons are usually methionines. It doesn't matter how many methionines it has within the sequence. That is really unimportant. Okay? So if I just take away the start codons again and leave the stops, you can see that there's no stop codon here. And then if I put back the start codons, you can see there are many, many methionines including this one here that is right at the beginning of the open reading frame. So again, a good chance that this will be a gene. However, all this we've been talking about start and stop codons is not answering the question of what is non-coding sequence. To answer that question, I'm going to take away the start codons again, taking them away. Okay, this is an open reading frame. Okay, if you look at the second reading frame below the first one that we were looking at, the open reading frame, you'll see that there's actually a short open reading frame here. Okay, so there could be a gene here as well. When we talk about non-coding sequence, we are looking for sequence where there are no open reading frames. Oh, no, I'm wrong. There are no genes. Okay, not every open reading frame is a gene. Many open reading frames are genes because they have a stop codon at the end, a start codon at the beginning, and a way to be transcribed. That's the third criterion. Sometimes the reading frame may just be open, but there's no way to transcribe it. There's no start codon, so it's not a gene. In that case, it's non-coding sequence. And sometimes you see sequence that has stop codons in all three reading frames, and then you know that that sequence cannot be coding. Okay, let me see if I can see one, or maybe if I move along, I'll see one. Unfortunately, this is bacterial DNA. So as I mentioned before, most bacterial DNA codes. But if this were eukaryotic DNA, what we're looking for now would be very easy to find. Okay? So I've highlighted a region that in all 6 reading frames, there are many, many stop codons. So that region is unlikely to include a gene unless that gene is very tiny. And that is non-coding sequence. It could have a range of functions. Okay, it could encode an RNA that is functional. It could be regulatory sequence that allows a gene next to it to be transcribed. You can see there's an open reading frame nearby. Maybe that is the promoter of that gene. Now, in order to tell for sure whether our open reading frames are genes and what the functions of the non-coding sequences are, we actually need to get into a lab and do wet lab work. But there's a lot that we can hypothesize just by looking at sequences online, looking for where the stop codons are, where the start codons are, and other features that will tell us whether or not something is a gene. Okay. Any more questions?
Okay, we have one from Adib. He's asking, what is the minimum length? What is the minimum length for us to call a chunk of DNA an open reading frame?
Well, obviously you have to have a peptide produced from it. Okay, and theoretically a peptide would need to be at least 2 amino acids joined together. Right? But in actual fact, most genes, in order to be functional, are bigger than 100 base pairs. Most, not all. So very often, when bioinformaticians are trying to, like, if a bioinformatician sequences a genome now and gets raw DNA, the raw DNA will look like this. I can ask software to translate the raw DNA in all 3 reading frames, tell me where the stop codons are. And then my next question is, where are the genes? The very first thing that somebody would do manually if they're looking for genes is to ask, please show me all the open reading frames. Okay, so let me do that. I'm asking the software to mark open reading frames, mark all the open reading frames. And the software is asking me back, what's the minimum size that I want it to mark? So this answers a big question. Other than 2, there's really no minimum. But most genes are bigger than 100 bases. So if I tell it to mark 100 and above, safely I should mark most of the genes. Some of the things that it marks that are 104 nucleotides will actually not be a gene. And there'll be some things that are 97 nucleotides that it will not mark because I did not tell it to mark, that are actually genes. Okay, there's been an instance where we were working with a plasmid and there was a small piece of DNA that we thought would have no function. And it ended up having a function. And I wondered why. And I realized that the gene is very short. So the people who had annotated it did not recognize it as a gene. Since we're this far, I'm going to actually mark all open reading frames that are 100 bases or more. Okay, it's done it. The beauty of being able to do things on computers is, if you think about it, I could have marked these manually with pencil on paper. It's just that if I did that for the 4.8 million base pair genome of Salmonella typhi, between now and my retirement, I would not finish. But I gave the computer instructions on what I wanted, and it's marked them for me. Okay? Abib, was your question answered?
Yes.
Yes, ma'am.
Okay, thank you. Can we notice that the open reading frames that are marked here, some of them are going from left to right, and others are going from right to left?
Yes,
Yes,
ma'am.
ma, we can see them.
We notice that, and we should not be surprised by it, right?
Yes, ma.
Okay, because the top strand, obviously 5' to 3' in this direction, bottom strand in the opposite direction. Now, this is a bacterial genome. Bacterial genomes have some genes that are transcribed together in groups. Those genes are said to be in an operon. So they would only need a promoter ahead of one of those genes where RNA polymerase would sit. And then when RNA polymerase sits and starts transcribing, it will transcribe all the genes in the operon into one mRNA, and that one mRNA would go to the ribosome and be translated, in this case, into 3 different proteins. Okay, operons are very common in bacteria, occasionally seen in plants, and very rare in eukaryotes, in other eukaryotes like animals, I mean. Okay, so this is a bacterial genome. And in bacterial genomes, the genes are very close together. Some genes are overlapping. What I have marked here are not genes. I've marked open reading frames. And you can see that they're close together and they're overlapping. But for me to be sure that they're genes, I would need to ask, is there a promoter upstream of them? And in the lab, can they be transcribed? So the way to find out where they can be transcribed in a lab is to find out whether you can isolate mRNA that has the same sequence as the gene. Okay? So when bioinformaticians are annotating, they'll take many factors into consideration before they call an open reading frame a gene. If I put back our start codons, I put them back, I want to show you a few genes. A few open reading frames. I want us to look into them closely. Here's the first one. Okay. I've highlighted it. By the way, I can highlight something on the top strand, on the top view, and it will also show in the letters. Okay. This is an open reading frame because there was no stop codon within it. But can we also see that there's also no start codon within it? So it's pretty unlikely that's a gene. Can we see that? There's no pink line inside it. I'll stop highlighting it so that you can verify, okay?
Sorry, ma, please. I didn't get that. Please, can you recap, ma? Sorry.
Okay. This open reading frame here that I've highlighted from 321 to 632. It's an open reading frame because there's no stop codon within it. But is it a gene? I now put in the start codons. And hopefully what you can see is that there's no start codon within it at all. So it's very unlikely it's a gene, even though it's an open reading frame. Does that make sense?
Yes,
Yes,
ma'am.
ma'am.
Yeah.
Okay.
Yes.
There's another one like it here. Okay. It's open, there's no stop in it, but there's also no start. Okay. This big one we were looking at from the beginning has many, many start codons within it. Any one of them could be the actual start. Maybe it's this one, maybe it's this one. Okay, but there's a good chance that this might be a gene. So there's a number of things we have to look at before we call an open reading frame a gene. Okay. Now it turns out that the chromosome of this Salmonella strain was sequenced in is it the 1990s or the 1990s. So it's been well studied. We know for the most part most of the genes in this genome. And the people who have figured out where these genes are have produced an annotation that allows me to look at where they have mapped the genes to be. So I'm going to take away our open reading frames for now. Take away our stop codons because they are start codons because they are making it busy, or even take away the stop codons as well. Okay. And I'm going to import an annotation that was done by people who've studied the genome. Okay. So I'm going to read an entry into the DNA file. And where is my annotation? That's my annotation. Okay, I have to clear some errors. There we go. Let's clear all the errors. Sorry. Okay, there we go. So here, these genes have been studied. Some of the open reading frames we looked at have been validated as genes, but not all of them.
Ma'am.
Pardon?
Now, please, I want to clarify something because it seems as if I'm mixing some things up. You said for it to be an open reading frame. Sorry, a gene may not be an open reading frame if there is a start and stop codon within it, or just a start codon within it.
None of those is an open reading. Sorry, the definition of an open reading frame is there's no stop codon. Stop. Okay.
Stop.
Yes. Just a minute. Let me see. Now all we have is stop codons, nothing else. Okay. An open reading frame. This is an open reading frame here. There's no stop codon within it. Can you see? That's an open reading frame. The start codons are methionines. Some methionines are actual start codons. Other methionines are just within a protein. So if I tell it to mark start codons, it's not really marking start codons, it's marking all methionines. Some of those methionines could be inside an open reading frame, and that is fine. Okay? So if I mark my open reading frames, you can see that thing I highlighted is an open reading frame. It doesn't have a it doesn't have a stop codon within it. Okay? If I mark genes, that part I highlighted is also a gene. But here, down here, we have an open reading frame that is not a gene. Okay? It was never validated as a gene. Any more questions?
Okay, yes, we have one from Bola Tito. She's asking What is a promoter?
Okay, a promoter is the sequence upstream of a gene or operon that RNA polymerase binds to be able to transcribe it. Any other questions?
Okay, yes, we have one from Adams here. He's asking, do you mean stop codon will not be present in an open reading frame at all, be it within or at the end?
It will not. We're defining an open reading frame as a piece of DNA that does not have a stop codon. That's the definition of an open reading frame. Okay, so if we find a stop codon, then it is not an open reading frame. And the reason why we're trying to define open reading frames is we're trying to understand whether a piece of DNA encodes a protein. If it has a stop codon in the middle of it, that means that its translation is stopped, so it won't make a protein. So if it doesn't have a stop codon in it, it could maybe make a protein if there's also a start codon and a place for transcription to start ahead of the start codon, the promoter. Does that answer the question?
Sorry, ma. What of adding of the open reading frame? What if there's a stop codon? Can we still say it is an open reading frame?
Sorry, can you ask the question again?
Yes, ma. I said, what about at the end of the open reading frame? If there is a stop codon at the end, is it still an open reading frame?
Every open reading frame has a stop codon at the end. Does that make sense?
Yes, ma.
So, this is an open reading frame. It starts here and it ends when I get to a stop codon. This is an open reading frame. It starts here, it ends when I get to a stop codon. I cannot say this is an open reading frame because there's a stop codon in the middle. But this is an open reading frame up until here, and right after here is another open reading frame. In fact, right next to it here is a very tiny open reading frame. The stop codons are defining what an open reading frame is.
Yes, ma. I want to ask a question there, ma.
Yes.
Yes, ma. So, in the open reading frame, so we have a start codon and a stop codon ending it. We have in the open reading frame, we have all through, we have start codon. Why ending it, we have the stop codon. So does that signify a gene? And you said before a sequence of bases can be recognized as a gene, it must begin with a promoter, like a promoter to initiate the something. So I want to ask that, how do you identify the promoter in these reading frames? Thank you.
Okay, let's go back, because we don't use start codons to define an open reading frame. We use stop codons. If there's no stop codon, it's an open reading frame. If it's an open reading frame, it may or may not be a gene. If it is a gene, it is an open reading frame because there's no stop codon within it, but it has additional features. It will have a start codon, which will usually be methionine. And it will have a way upstream of that gene of being transcribed, typically a promoter. Promoters are short sequences that RNA polymerase can bind to. Sometimes we can recognize them because RNA polymerase has favored short sequences that it sticks to. But sometimes they're difficult to recognize because those favored short sequences are not there. Sometimes RNA polymerase does not bind on its own. Another protein helps it to bind. So sometimes if I can't see a clear if I can't see the clear sequences that RNA polymerase often binds to, it doesn't mean there's no promoter. It may be that there's another protein that has its own binding site there, and that protein binds RNA polymerase. So what am I saying? Looking for promoters is far more complex than looking for open reading frames. The signals are not as clear and simple and straightforward. Does that answer your question?
Yes, ma. Yes, ma. Thank you.
Okay. Bear in mind that in the bacterial genome, not every gene will have a promoter because some genes are in operons. A gene could be transcribed because it has a promoter right upstream of it, or because of a promoter that is far upstream and is helping to transcribe other genes at the same time. Okay, here we have genes of Salmonella typhi as annotated by scientists that show some genes very close to each other, likely in an operon. They also show that you can have genes on either strand. So this question came up earlier. So for this gene, the top strand is the coding strand and the bottom strand is the template strand. But for this gene, it's the other way around because it's going to be transcribed in the other direction. In bacteria, I mentioned sometimes the genes are overlapping. Let me see if I can find some. You saw open reading frames that were overlapping, but notice that many of them did not turn out to be validated as genes. But there are a few genes in bacteria that actually do overlap. Okay, let me see if I can see one quickly. I saw one earlier, but I thought I could find a better, clearer overlap. And overlapping genes can overlap on the same strand or they can overlap on different strands. Not seeing any. But believe me, they exist. Okay? And hopefully when you start doing your own bioinformatics, you'll see a few. Okay, if you're pharmacy students and you're on the pharmacy PharmD students, you'll learn to be able to work with DNA on the software that I'm working with right now in your genomics class in part 5. Okay. Maybe I need to give them an assignment to find me overlapping genes, and then I can show you guys in bioinfo. Okay, I'm going to close the Salmonella window. Are there any questions before I do that? And then when I do that, I'm going to open a portion of a genome of a eukaryote. Any questions?
Yes, ma. I have a question.
Okay.
I wanted to ask about sigma factor. So,
Yeah.
I mean, sigma factor is used sometimes to bind. Does sigma factor have its own specific usual binding site, or it's random? So, is there like a sequence it usually binds to?
Yes.
So it can bind to. Okay.
Yeah. So to bring everybody up to speed on your question, can you tell them what sigma factor is?
So sigma factor. So earlier you were talking about how sometimes promoters require some help locating. I'm sorry, the tRNA requires some help. Sorry, the polymerase requires some help locating where to bind on the sequence. So sometimes it doesn't find the promoter easily. It uses some proteins. One of those proteins is called sigma.
No, wrong definition. Sigma is part of RNA polymerase. Okay, let me just call up the slide. Give me a few minutes. Where would this be? Where would this be? Where would this be? PHM.
Oh, I think I got it. Sorry, I think sigma factor is one of the subunits
It's one of the subunits of RNA polymerase.
in
Yeah.
the
So
RNA polymerase. Yes.
I would like to actually get a slide and take us through this properly, so that people don't get confused. And then I can also explain what I was talking about, other proteins helping and how that is different. Okay. PHM. Oh, my computer wants to crash. I'm opening too many things. What can I close? No, I can't close that. Just a minute. Okay, let's close Excel. Okay, so I'm looking for, the trouble is I PHM 710. I think I tossed it in 710 once. Just a minute. Okay, Google Meet. Stop presenting. Share. Share window. Found the PDF it will have to do. Okay. This is a picture. Can we see the slide? This is a picture of RNA polymerase. Okay. And the RNA polymerase has different subunits. There are the alpha and beta subunits, which you can see in blue and brown and so on. They have different parts of this. Okay, they are the ones that actually do the transcribing. Okay, and there is the sigma subunit. It's a part of RNA polymerase. Sigma has sequences that it likes to bind to. We often say that sigma has 2 feet. One foot is put down at the -10 position. +1 is where transcription is going to start. +1 will be right ahead of the start codon. Okay? At -10, sigma will put down 1 foot. And if it's this particular sigma, it likes to step on TATAAT. Okay? And then it will put its other foot on the -35. It likes to step on TTGACA. If it sees those sequences, it will sit or step very comfortably. Okay? It turns out that very few genes at the -10 position have TAAT and have this sequence at the -35. Most genes will have some of these nucleotides and not all. For example, at -10, maybe it has TGGAAT. That's enough for sigma to step on. It won't be stepping as firmly as if it stepped on TATAAT. And in the same vein, in the -35, maybe it has TTCCCA instead of TTGACA. Again, it will step well enough. Once sigma binds well enough, it will recruit the other subunits. And those subunits will then start transcribing. Sigmas is a subunit of RNA polymerase, but its only function is to pull RNA polymerase down onto the promoter. Once it's done that, it's finished its job. And very often, sigma will then leave and then go and look for another promoter. The reason why it leaves is that sigma tends to be in shorter supply compared to the other subunits, but it's still part of RNA polymerase. Okay? You will notice in this figure that there's another part of sigma, the sigma 3 part of it, that is looking for a D, a T, G, and any nucleotide to stand on. If that is there, it's helpful. If the -10 is not such a good fit and -35 is not such a good fit, but that middle is there, it will help sigma to be able to stand. You also notice that the alpha subunit, there's some things that it can bind to, but not as well as sigma. We call those the up element. If the sigma binding sites are not that good and the up element side is good, there's a good chance that RNA polymerase can start transcribing. Okay? Now, there are some promoters that don't have any good binding site for any of these -10, -35, the sigma 3 binding site, the up element binding site, they're not there. But there may be another protein, not RNA polymerase now, but a completely different protein that is able to bind there and also bind sigma. If it can draw sigma to the promoter. Sigma will bring its other subunits. Transcription will start, even though there's no sigma binding site. So this is why I said it's very difficult to find promoters because they're never perfect. It's not absolute like the genetic code. Okay, when I see UUU, it's phenylalanine. With promoters, it's not that way. It's a maybe thing. So you need a lot more computational power and intelligence to be able to find a promoter. Interestingly, AI now has increased our capacity to find promoters because it needs some intelligence, actual intelligence from human beings who can read experiments or artificial intelligence. But sigma is part of RNA polymerase, and there are other promoters that can help sigma to bind. Now, one more thing about sigma, because we are here, there's not one sigma. There are different sigmas, okay? So the sigma we're looking at now is called sigma 70. They're named by their weight. It is the sigma that does, and by the way, the sigmas here are the sigmas in E. coli, which are the best studied. Sigma 70 does most of the transcription in E. coli. Okay? But when E. coli grows to stationary phase, sigma 70 becomes in short supply, and then other sigmas, stationary phase sigma, becomes more common. That's what's not even on my table. And then when the bacteria are heat shocked, sigma 70 is destroyed, and sigma 32 is transcribed better. It turns out that many of the genes that have good sigma 32 binding sites are genes that can help the bacterium to respond to a heat shock. So it's one of the ways that the bacterium chooses what to transcribe. None of this is what I conventionally teach in Module 0, but it's something that is important to understand ultimately if you want to be a molecular biologist or a bioinformatician. So I thought I should respond to the question fully. Is the question answered? Is this clear?
Yes, it is. Thank you.
Yes, ma'am.
Okay.
Thank you, ma'am.
All right, so let's take this away. And where's Google Meet now? Stop presenting. And then, oh, I was even going to open us a new piece of DNA. So let me do that first. I was going to open us a eukaryote. So you would think that, well, I showed you Salmonella DNA. Actually, normally what I show is E. coli DNA because E. coli is my best friend. But I had Salmonella handy because I used it to teach yesterday. And you would think that being a human being, that I would then show you perhaps human DNA. Unfortunately, I have no interest in human DNA. I don't have any on my computer. So we're going to look at DNA from Plasmodium. Okay. Plasmodium is the parasite that causes malaria. Okay, so let's share my screen. So I've opened a piece of DNA from Plasmodium nowelski, which you may not have heard of because it doesn't cause DNA. It doesn't cause malaria in humans. It causes DNA. Causes malaria in mice. But what we just want to do is look at DNA. Okay. So same software. So we're looking at the DNA strand, 5' to 3'. This is our top strand in letters, bottom strand in letters, all 3 reading frames for the top strand above it, all 3 reading frames for the bottom strand below it. And what you have on top is the same thing you have at the bottom, but zoomed out so that you can see more. Okay. And hopefully one of the things that you will notice is that the genes are much further apart. Can we see that? There's a lot of space between genes. So the question about non-coding sequence, all of this sequence here is non-coding. What does it do? I don't know. A malaria biologist might know, but it does not encode a protein. Okay? Now I want us to keep looking, and you'll see a gene here or 2 genes here. One of them is called FAT4, and the other one is called FAT5. Hopefully, you can see that FAT4 starts here. I've highlighted it. It starts here on this reading frame. There's a pause in which there are stop codons. And then it continues on another reading frame. There's a pause with stop codons, and it continues on another reading frame, and so on. There are little pieces of FAT4 that are coding that are separated by pieces of FAT4 that are non-coding. If we look at FAT5, same thing. FAT5 has only 2 pieces. They're separated by a piece in between. What are these pieces in between that are non-coding, that are between parts of the gene?
Exon.
Pardon? Someone spoke. I didn't hear what the person said.
I guess exon.
No, they are not exons. The exons are the parts that are coding. So this part here that is in an open reading frame, this part here, this part here, this part here, in FAT5, this part and this part are all exons. The parts between them are not exons. What are they?
Introns.
They're introns. Thank you. So introns are non-coding pieces of DNA that are in between coding pieces of DNA that are referred to as exons. So this DNA, when it is transcribed, the mRNA will start from here and end all the way there. And then when the mRNA is processed inside the nucleus, the introns will be removed and the exons will be spliced together. And it's that spliced version that will now be exported through the nuclear membrane and will go to a ribosome to be translated. I'm sure we learned this at some point. But I hope you can see that when we say that eukaryotes have a lot of non-coding DNA, some of that non-coding DNA, like this, is outside of genes. And some of the non-coding DNA is inside the gene. Any questions? So the thing about splicing that makes it even more complicated is that sometimes there can be a gene that encodes more than one protein because that gene can be spliced in different ways. So you notice that in this figure, we have FAT4, which is highlighted here, with 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11 exons. But there's something below it highlighted on the DNA sequence called FAT4 alternative. That means that there's an alternative protein that can be made that has 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 exons. The proteins are similar, but they're not the same. So FAT4 will be a slightly longer protein. No, it will be shorter actually because there's an intron here. And FAT4 alternative will be slightly longer. And they could have similar or different functions. So even though the eukaryotic genome, less of it is coding, it can make more proteins because the coding sequence can be reused in very creative ways. Does this make sense? Any questions?
Sorry, hello, ma.
Yes.
I thought when you clicked on the blue sequence
Mm-hmm.
or the blue, I thought it was explaining like the red and the blue. I thought they were similar. So why
Yeah.
is this one and not? Are they not supposed to be? When you click on it now, I was thinking it's supposed to be explaining or similar to the red. So
Yeah.
now the fact that alternatives means that they are separate or were they rearranged?
No, so on the gray lines, you have DNA sequence. Okay. And on the reading frames, you have the translated amino acid sequence. Normally, we shouldn't need the red. You notice that for FAT5, there's no red. For FAT6, there's no red. Okay? The reason why there's a red for FAT4 is that FAT4 has different ways of defining its introns. In FAT4, this region here can be an intron or it can be part of an exon, depending on how the RNA is spliced. So the RNA could be spliced in such a way that this region here is removed or it's left intact. And if it's left intact, you make a different protein. So in order to show me this in my same window with my same software, the annotator has marked FAT4 as he or she believes it should be spliced, and an alternative FAT4, which is spliced in a different way. Does that make sense?
Yes, ma.
Yeah, it would have been possible to put the FAT4 alternative on here as well. It's just that they'll be on top of each other and I won't be able to see them. In some cases, the alternatives are many. Sometimes there are 10 splice variants. In that case, I'll have to load different annotations to be able to see them. Okay? Okay, let's just quick. So this is just a portion of the plasmodium chromosome. You see there are large parts of it between genes that are not translated. Okay, and this is what in the past people used to call junk sequence, but which we now know has other functions. Okay. Here's another gene with an intron. Okay, so in plasmodium, I think roughly half of the genes have introns. In humans, almost all the genes have introns, okay? This is a gene on the reverse strand that has introns. Okay, FAT17. I think that's the end of the sequence. Okay, so you have a picture of what a bacterial genome looks like. You have a picture of what a eukaryotic genome looks like. Viral genomes will typically look a lot more like a bacterial genome. They typically do not have introns. But what is very, very common in viruses and much less common in bacteria is having genes that overlap. Viral genomes are small, and it's very common for them to have genes that overlap and very, very little non-coding sequence. Okay, any questions on today or on last Friday?
So we have a couple of questions here on the chat box. Okay, Clemens, you can go first with what you want to ask. Then I will read from the chat box.
Yeah, my question is that when you talk about the open release frame, you said you have 6 open release frames. So if we start from the forward strand, we have +1, +2, +3. And we start from the first strand, we have -3, -2, -1. So I've worked with a DNA strand of E. coli polymerase 1. So when I look for the open release frame, the +1 open release frame, when it does the open release frame cycle, so I later found out that the one that has +1 has the longest open release frame. Does it always be like that?
No. No. So, I mean, we're talking about DNA polymerase. I just want to make something clear in case anybody else. We don't need reading frames to replicate DNA. We don't need the reading frames because we're just dealing with the nucleotide. But with translation, reading frames become important because we're now looking at codons, and which which reading frame you have depends on where you start. Any reading frame can have any big genes, small genes, and so on. In fact, which one is on top depends on where you start reading. For bacterial genomes, they are circular. Okay, this is plasmodium, so it's a eukaryote. Bacterial genomes are circular, so that we're reading them on my screen, which is not circular, is because we have cut the sequence at a particular point and started reading from that point. But I could cut it anywhere. I could start reading it from anywhere, and that would determine what is on top and what is second and what is third. Okay? Some of these things that we're using to visualize are not inside the cell. We have 3 reading frames, and we are calling 1 +1 +2 and +3 because of where we started from. Okay, so it's not that any particular reading frame has bigger genes. The only thing to pay attention to is that the reading frames on the top strand are going to be reading in this direction, and on the bottom strand are going to be reading in the other direction. Okay. Tochi, I thought you said there are other questions.
Yeah, I'm about to read them. Okay, so we have one from Michael here. So he's asking how splicing is done. And a follow-up, is a splicing enzyme in a microorganism, or we have them in the lab?
Okay, splicing is done inside the nucleus of a eukaryotic cell. There's a collection of enzymes that are referred to as a spliceosome. And they form a kind of loop. They make the intron form a kind of loop. and then they catalyze the removal of that loop and the joining together of the two exons. Okay, if you want more detail than that, I'll suggest get a eukaryotic molecular genetics textbook. They describe it very well. Some of them are, if you get molecular biology of the cell through NCBI books, you get an edition there. It will describe it very well for you.
Okay, thank you. So Joy and Adams have similar questions. So they're asking about the functions of introns beyond splicing out, and if there are any, can you give examples of them?
The functions of introns are really not known. Now, saying that, bear in mind that the person who is saying that is somebody who studies bacterial genomes where we don't have introns. So I'm not up to date on the intron literature. Maybe last year somebody discovered the true function of introns, but by and large, they're unknown. And there are a lot of people who study splicing and spliceosomes who are trying to figure out what exactly is the function of introns, because it sounds very wasteful to make a long RNA and then remove parts of it that you're not going to use. So why that happens in eukaryotes is not really clear.
Okay.
Actually, let me just add something to that answer. While I cannot answer what the functions of introns are, somebody asked earlier how you find promoters. It's actually very hard to look at sequence and find introns. It's again another problem that AI is trying to tackle. Most of the time, when introns are marked up, they're marked up because somebody did an experiment to find out what the introns are. And the way you do that is you take RNA after it has been spliced, you reverse transcribe it back to DNA, and then you sequence it and find out what is missing. And then you know that was what was removed by splicing. Okay.
Okay, yeah, thank you, ma. I think that's all we have for now.
Okay.
But if you have any, you can unmute and ask, or you can see Jordan. We have just a couple of minutes to do that. Okay, Abib, you can go ahead.
So, okay, so regarding the intron question, so, correct me if I'm wrong. So I'm thinking they might actually be beneficial because, we know that for every cell division, so there will be probably error in DNA replication. So most of the time, so, the mutation, most likely they are going to fall within probably the introns. So in a way, I can say probably introns might actually be beneficial because, majority of the mutations that we have, so most likely it's going to fall in the junks or the introns. Thereby, I'm finding some advantage in it.
Yeah, I think that's true. A lot of mutations fall in introns and are non-consequential. I did, however, want to point out that some mutations that occur in introns change the way the introns are spliced.
Yeah.
On the one hand, this does, it can potentially be deleterious, but on the other hand, it's the way that new splice variants evolve. So I completely agree with you. Introns could have that function, but all other non-coding sequence has that function as well. So the question becomes, why are introns there and then removed? It's a partial answer.
Okay.
Thank you. Any other questions?
I guess that's all we have for now.
Okay. All right. I think we have just one more module 0 session after this, and then we should be done.
Okay. Thank you very much, ma.
Okay.
Yeah, sorry, please, I want to ask, how can we familiarize ourselves with all these things we have been taught today? Like, is there any material or YouTube, something apart from that of genetic code, especially these reading frames, how we familiarize ourselves with all you have taught us on it? Thank you.
Yeah. So, first of all, I will send you the paper on genetic code in addition to getting yourself the genetic code. And then secondly, I have made some videos teaching this. Okay. So I will send you links to those videos. Okay.
Oh,
So
thank you,
all
ma.
this stuff about reading frames and so on, if you want to go over it, you can use the videos to go over it. And then hopefully that should be helpful. If there are other questions after you've looked at that material, bring it to the next class. And of course, I'll share the slides. Adam said some of us are finding it difficult. There are many, many, many genetic code apps. So if one is giving you trouble, please just uninstall it, throw it away, and download another one. Yeah. And don't pay for them because there's many, many that are free. Okay. There's a question: do open reading frames encode proteins? Those that encode proteins are genes. So when we start talking about open reading frames, what we're really looking towards is this a gene? Not all genes encode proteins, by the way. Some genes encode proteins, some genes encode RNAs. But when we find an open reading frame, our next hypothesis is, does this encode protein? And that's why then we start asking, well, is there a start codon as well as a stop codon? Is there a promoter that this thing could be transcribed from? And ultimately, we will ask in the lab, is a protein made from this? Okay. People are asking about Artemis. You can actually download Artemis and use it. If you want to do that, I suggest some instruction. You could probably figure it out on your own. A few people in the 5 class said they figured it out on their own. But I'd suggest asking someone who knows how to use it so that you don't spend a lot of time trying to figure out how to open the files. Artemis is a computer software. It's best to run actually on Linux, right? But I am running on a Mac. You can run it on a PC and use it as a sequence browser. It's free, so you can download it from the GitHub online. But as far as I know, there's no mobile version. And partly because you probably wouldn't want a whole genome on your phone. Okay. All right, I think we have to stop there. Happy to take more questions next time. Thank you all. Bye.
Yeah, thank you very much, Prof. Thank you.
Thank you, ma.
Bye.
Yeah. So see you guys next week, Friday, same time.
Hi,
Thank you
hello.
very much.
Oh no. God, I beg.
Hi, good evening.
Yeah, hello, good
Hello.
evening. Yeah, I can hear you.
Please, is the test going to be next week?
The test, no, we still have one more class before the test. So after the
Oh,
test, then probably the week after it, yes, would have
okay,
the
the
test.
week after, right?
Yes.
Because we have our exam next week. Sorry,
Yeah.
test rather.
Okay, next
We
week.
might not.
Exam?
Yeah.
What department?
Sorry, test.
Oh, okay,
Okay.
okay.
Is it clashing with the time for the class? That is 4 p.m.
Okay, so not like the module 0 exam will be next week Saturday, right? I just want to confirm that.
Okay, no, not at all. Not at all.
Thank you.
Yeah.
I'm sorry, good evening.
Yeah, hello,
Can you
good
hear
evening.
me?
Good evening. No.
Yes, I can hear.
Next week Friday's lecture will clash with our test. We have tests from 3 to 5. Yes,
Okay.
PCY. Yes, we have PCY 5113 from 3 to 5. So it's
Okay.
going to clash with, yes, for pharmacy students. We have tests on Friday.
Okay. We would have to get back to Prof to see if we can make it 5 p.m. Otherwise, we might have to release the recordings for you guys afterwards. Yeah.
Okay. Thank you very much.
Yeah. Thanks. Is any other questions?
Please, I have a question, sir. Please, I want to ask if there will be AOC for for the test that will be coming up.
Is that okay, Victor? For now, you just have to go with whatever prof is teaching for now. So, yeah.
I have a question.
You have a
Okay,
question?
okay.
Okay.
Thank you, sir.
Yeah.
Hello, sir. Please, I would like to ask, is it going to be a physical exam?
Yeah, hello.
Is it going to be physical, the test? Is it going to be a physical test?
Yes, it's going to be a physical test. So communicate the venue to you guys towards the date.
Okay, thank you.
Yes, thank you.
Hello, sir. I have a question.
Yeah, but let's do, yeah, good evening. You can go ahead.
The test, is it going to be a written test or like pen and paper test or with?
Yes, it will be handwritten, objective, subjective, and small theory. Yes.
Okay.
Thank you.